Hello everyone, our dear senior tools, you are most welcome in this lesson 3 in our topic transport of materials. Then in this topic, expect to define the term osmosis, to explain how osmosis occurs, to demonstrate the process of osmosis in living tissue and artificial membrane, to explain the terms used and other expectations. To start right away, when you're talking about osmosis, we are talking about the movement of water molecules from their region of high concentration to their region of low concentration across a semi-permeable membrane, i.e., water molecules move from dilute to a concentrated solution, because there are very many in a dilute solution and very few in a concentrated solution across a semi-permeable membrane, and the semi-permeable membrane is a porous membrane, i.e., it has some pores which allow some materials or molecules to pass through and prevents others. To demonstrate osmosis here, we have two solutions. We have sugar solution and then water. Then molecules are moving from sugar solution to pure water by osmosis across a semi-permeable membrane, as demonstrated here. Check. Then experiment to demonstrate osmosis in an artificial membrane. Here we use a whisking tube, and then we organize our setup as shown here. We have sugar solution in the whisking tube and then water in the beaker. Then after 30 minutes, the solution in the capillary tube rises due to absorption of water from the surrounding solution by osmosis. The whisking tube absorbs water by osmosis. Then experiment to demonstrate osmosis in living tissue. We have here, we need Irish potato cut into a cup shape, and 3 of them, A, B, and C. In A, you put some sugar crystals. In B, it's empty. In C, it's a boiled cup with sugar crystals. Then after 1 hour, there is a rise in sugar solution in A, and then a decrease in the petri dish. Then there is no sugar solution, or there is no solution in B, and then in C there is solution at the same level with that solution in the petri dish, implying that C is fully permeable. Then B, there is no osmosis because there are no sugar crystals to attract the water molecules. Then in A, the sugar solution absorbs water by osmosis from the surrounding petri dish. That is to demonstrate osmosis. Then terms used in osmosis, we have hypotonic. Here we can use a more dilute solution. We use these terms in comparative terms. More dilute compared to the surrounding solution. If the two solutions are surrounded by a semi-permeable, are separated by a semi-permeable membrane. Isotonic, same concentration as the surrounding solution. Hypertonic, more concentrated than the surrounding solution. Remember, we said water molecules move from dilute to a concentrated solution. Then water potential, we are talking about the tendency of water molecules to move, or the ability of water molecules to move. There, this is what we call the water potential. Potential is the ability. Then solute potential is the tendency of reducing the ability of water molecules to move. If you add the solute into a solution, you are reducing the ability of the water molecules to move away from that solution to another solution if they are separated by a semi-permeable membrane. That is solute potential. Then to demonstrate osmosis in animal cells, here we use red blood cells. if you get a red blood cell and place it in a dilute solution, in a more dilute solution than the cytoplasm of the red blood cell, the red blood cell absorbs water by osmosis and expands beyond the maximum size and then bursts, a process known as hemolysis, which is the bursting of the red blood cell as a result of absorption of excess water by osmosis. Then, if you get a red blood cell and then place it in a concentrated solution, its cytoplasm is going to lose water to the surrounding concentrated solution by osmosis. As it is losing water, the cell is going to shrink, a process known as chlorination. Chlorination as the process. Then osmosis in plant cells. Unlike animal cells, plant cells do not burst because they are surrounded by a rigid cell wall. So in this case, if you get a plant cell and place it in a dilute solution, the plant cell is going to absorb water by osmosis. Its vacuole and the cytoplasm will expand and exerts pressure onto the cell wall, a pressure known as Tagger pressure. And then the cell becomes turgid, a process known as turgidity as a result of absorption of water by osmosis. Then, if you get a plant cell and place it in a concentrated solution, this plant cell is going to lose water to its vacuole, and the cytoplasm is going to lose water to the surrounding concentrated solution by osmosis. The cytoplasm and the vacuole shrinks away from the cell wall, causing the cell to become flaccid, a process known as plasmolysis, the shrinking of a plant cell as a result of losing water to the surrounding concentrated solution by osmosis. Compare it with wilting. Wilting is the shrinking of a plant cell as it is losing water by evaporation, whereas plasmolysis is by osmosis. Then, importance or significance of osmosis in plants is very important as it results into absorption of water from the soil, absorption of water only from the soil. Then it facilitates the opening and closing of the stomata as when the guard cells either absorb water to expand or lose water to shrink. Then it facilitates support in a non-woody plant as a result of absorbing water and become rigid. Then movement of water from the root air through the cortex up to the xylem is also by osmosis. Then in animals, movement of water in and out of the unicellular organism is by osmosis. Absorption of water from the gut, the colon, is also by osmosis. Reabsorption of water from the kidney tubule, it is also by osmosis. And then movement of water from the tissue fluid into the tissue is also by osmosis. So it's a very important role. Then for more information, please check our e-learning platform to get the notes, and then you compare with the explanation to get the detailed lesson. Otherwise, I wish you the best. Thank you.